Hi everyone, my name is Pierre Poulain. I'm a lecturer at University Paris Diderot. And yes, my talk today is about using Python to fight malaria, but also trying to explain you why open science is important in research. This work has been made possible by my employer, of course, my host institution, CNRS, and the strong partner we have in the Republic of Congo, the Congolese Foundation for Medical Research. So malaria is a mosquito-borne disease. It means you get malaria from a mosquito bite. Actually, not all mosquito can give you malaria, only the Anopheles type of mosquito, only the female, actually. So during her blood meal, the mosquito will also give you a little gift. This is a parasite from the malaria. This is the one responsible for the disease. This little guy here is very small. There is different kind of parasites. The name is Plasmodium. And once you get infected, after a few days, you're gonna feel sick with fever, headache, chills, sometimes diarrhea, and eventually you can die from this. Actually, if you have been infected with Plasmodium falciparum. Malaria killed actually in 2015 more than 400,000 people worldwide, and it has also infected more than 200 million people. The burden of malaria on developing countries is very heavy because once you get infected, when you get the disease, you eventually get diagnosed, you get treated. This is the good news, but you are totally out of order for several days. And when you are sick, you cannot go to school, and also you cannot go to work. You find malaria basically in South America, Asia, and mainly sub-Saharan Africa, where Africa actually accounts for more than 90% of all cases worldwide. So, diagnostic, you can diagnose malaria with different ways. The most common and spread one is optical microscopy. It just means you need a microscope to diagnose malaria. There's two kinds of examination you can do. It's a thick blood smear. So this is a glass blade for microscopy examination here. And the thick blood smear is a drop of blood you put onto the blade, and you add a reagent, let it dry, you wait for a while, and then you can look for parasites at the microscope to this thick blood smear. The other one is the thin blood smear. This is roughly the same thing, whereas here you put some blood, but you're gonna spread it into the blade. And the most common examination is a thick blood smear. This one is used for specific cases when you want to know exactly which kind of parasites you have into your blood at which stage of development. So this is optical microscopy is the gold standard for malaria diagnosis. This is the examination recommended by WHO. This is a very tedious task. It takes more than 1 hour for one technician to prepare the sample and then to look for parasites with the microscope. And the technician needs to be very skilled. This is very difficult to find parasites. I'm going to show you some examples here. So looking for parasite is in a thin blood smear. This is the easiest case here. So the pink bubbles here are red blood cells, and you have the parasite responsible for malaria as purple things, ring shape here, and I've highlighted them with a green box, as you can see. This is very easy. And even harder case is this one. This is a thick blood smear. This is the most common kind of examination you have to perform to diagnose malaria. You don't have the red blood cells now because they've been destroyed the preparation process. You have the white blood cells here and here. And the parasites are here, 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 also here. But I might have missed some of them. This is what, on everyday life, a technician has to do when you want to diagnose malaria. So, could technology help into finding parasites on the image? We believe so. Why? Because within the last few years, we had very nice development into image analysis. I really like this project by INRIA, I guess. It's called PlantNet. So you take a picture of flower with your smartphone, and then it will give you the name of the flower. So this is a very funny application, very interesting. 2 weeks ago, I came across on Twitter on this news saying that there was a new artificial intelligence that can tell if a sheep is in pain, based also on image analysis. Okay, good for him. But for human, we had a very nice paper published in a very famous scientific journal called Nature, saying that we are now able to take a picture of your skin and then tell you, with this kind of algorithm, tell you if either you have, whether or not you have skin cancer, but also which kind of skin cancer. So this is very nice, but I like to say, to stress on the fact that if you want to do such kind of algorithms, here this is deep neural networks, you need a lot and a lot of data. This is very important for machine learning techniques. Well, what about malaria? This is what we are interesting. So hopefully for us, about 2 years ago, we saw a journal, a review article published with a title saying that computational microscopic imaging for malaria parasite detection. So it was very good news for us. This is exactly what we want to do. Look at an example they did here. So you have a thin blood smear. This is where exactly you want to find parasites. The parasite is the little thing here. And with some kind of image analysis algorithm, you are able to spot exactly where the parasite here, exactly here. So this was a very, very, very good news for us. It was even better because in this same paper, the author counted the number of papers related to analysis of blood smear image in the context of malaria diagnosis. And altogether, this represents more than 100 papers between 2000 and 2015. So this is definitely very good news for us. However, we were not able to find any data sets. We were not able to get any source code. So we had to rebuild again and again. We cannot do such a thing. We need open data and open source code to be able to do proper research. You cannot spend time and money doing the same thing all over again. Our solution, that is just one solution among many others, of course, is kind of database, of web-based database called MalariaSmear Search. It has 2 different modules. The first one is the ability to upload blood smears and annotate parasites. There's no point to upload a picture for malaria diagnosis if you are not able to precisely annotate where exactly are the parasites, where are the red blood cell, where are the white blood cell, etc., etc. The other module is an e-learning module to train technicians to recognize correctly parasites. It's worth mentioning that before doing any machine learning with your dataset, it's worth spending some time trying to do some e-learning, because capacity building is very important in developing countries. Before going any further, I'd like to also to mention that most of the code of this project has been done by Athénaïs. She's a master student in bioinformatics working with me. And if you have questions, you can ask me or ask Athénaïs too. And of course, you can download all source code already available on this GitHub repo. So the backend is pretty common. We are using Flask. It's very nice to develop quickly some web. SQL Alchemy is used for the binding with the SQLite database. We don't want to become the next Facebook for blood smears. Okay, this is not the purpose here. So we use something very lightweight. It's worth mentioning that with Flask you can use a lot of different extensions. The one we used was Flask login for user authentication, Flask what's the form and uploads for dealing with HTML forms and image uploads, and Flask JS Glue. This is the black magic. This is because you can bind more easily your backend in Python with your front end in JavaScript. This is really black magic to me, but it's working very nice. The front end part is the usual trilogy with HTML, JavaScript, and CSS. Two very interesting JavaScript we used. Cropper.js, that is a JavaScript library to let you the ability to select very easily an area into a picture. So this is very convenient. And Canvas, .js, that is just an enhancement of the basic HTML5 canvas. So of course, here are a few screenshots of our app. Of course, once you have uploaded the picture and added all metadata, you can easily select an area of interest. For instance, I want to highlight this one here, it's a parasite. And then you can tell to the app to add the annotation which kind of parasite it is. Here it's a trophozoid. And you can also highlight a red blood cell, such as this one. You can, once you have annotated all your image, you can easily browse and navigate among all other annotations. You can highlight specific annotation. Here it's a red blood cell in yellow. If you are not happy with the kind of annotation you made, you can also change the annotation itself. Regarding the diagnosis of malaria, it's sometimes very difficult to make the difference between different stages of parasite and different kind of parasite. You need to be very skilled to do such a thing, so having the ability to change it on the fly is quite convenient here. As I said already, it's very interesting to spend some time into capacity building, and e-learning is a way to enhance capacity building. So we designed two different activities. The first one is we display a blank image. This is a thin blood smear. This is, again, the easiest things to do. And you have to spot exactly where are the parasites. Okay? If you made a mistake, you have red crosses here, and if you eat the correct parasite, you have the green boxes coming up. And so in this image, you have actually four parasites. You have one here, it's a ring stage, another one, so it's probably trophozoid, and another one hidden here. You have to do this within a limited amount of time, as any technician will have to do in real life. So you have to be quick and accurate. The other kind of activity we made this time we display a blank image, but we highlighted a specific annotation, a specific object within the image, and we asked the technician whether or not it's a parasite. You have to choose very quickly also and make fewer errors as possible. So far, most of the code have been developed. We're working with our colleagues at Brazzaville in the Republic of Congo. So at this time, they are recruiting patients and collecting blood samples. And they will prepare all the blood smear and, of course, taking picture with this kind of device. So it's just a regular microscope with on top of which is mounted a camera. And you can, of course, send all the picture into the database and add all the annotation regarding the image. A big issue we have in science nowadays is sustainability. In a couple of years from now, I have no idea if this project, if the server will be still up and running because of funding. I have no idea if I have be able to have money enough to spend for the server and the application and so on. So you already saw that the code is already on GitHub on a repo, but what about the data? Data are the most important thing we have. So we don't want to lose them. So we have decided to, each time we're gonna collect data, we're gonna transfer all this data to Zenodo. I don't know if some of you are very used to Zenodo. Zenodo is a kind of storage facility for research data. It's free. The money comes from Europe and the IT facility from the CERN. So it's pretty robust. And Zenodo implementing the FAIR principles. It means that your data will be findable, accessible, interoperable, and reusable. So this is very important for us. In order that the data won't be lost anymore. So we tried to perform some basic image analysis using Scikit image, OpenCV, to make the difference between parasites and the rest of the image, to highlight specifically the parasite. We failed. So if some of you are very good at image analysis, please come to see us. we have some challenge for you. And to end my talk, I'd like to, I borrowed actually with his courtesy 2 slides from Jake van der Play's keynote at PyCon in 2017. The first one is a quote by Isaac Newton, a very famous scientist, saying, "If I have seen further, it's by standing on the shoulders of giants." This is the usual scientific research we're doing in research. We always using previous work done by researcher. This I apply as well for Python code. If I've seen further, it's by importing from the code of giants. Of course, it's not. This is a modern version. And the takeover of this story is really, I'd like to thank you, the Python community, for bringing us scientists very nice tools, very robust tools, such as NumPy, Pandas for the analysis, Jupyter's Notebook, as you saw previously, but also Scikit-Image, Scikit-Learn, and so on. So, thank you.